Let's start second part. Second part includes merging of different groups up to the data normalization. In this video, we learn between this, we visualize our data, how is our data and how we need to merge different groups. Let's start. Now question is that why we need to merge. It's not necessary if we focus on individual group, but it is better if we compare two groups or two conditions. Just like in my case, I was comparing control and LPS group. This enables joint clustering, UMAP, PCA, etc. We can compare groups easily, and it is easily to tell us about the cell types between two groups. Before merging, we must label our data, just like control for control and LPS for LPS. Now let's merge. Just like before, there are four only: control, Seurat, and LPS Seurat. Now let's again combine. Combine indicates we give a name combine because it is a combination of control and LPS. Now if we look at the combined data, here is in feature and in count, and here is the group. Now you may confuse that where is the LPS. If we go down, there is the LPS. After that, second is quality control. For the quality control, we must I want to brief some terms which are mostly used in single-cell RNA sequence. One is N feature, which is also known or simply called as a gene per cell. Second one is N count, which is simply called as a molecules or UMI per cells. And third one is %MT, which indicates mitochondrial percent. Now let's we look at the mitochondrial percent. Mitochondrial percent is most important because it tells about the health of cells. If mitochondrial percent is too high, it indicates cells are under stress or apoptosis. Simply we can say cells are dying and poor quality cells. For this mitochondrial percent, we use this command. In background of this command, it works just like by using this formula: UMI counts from MT, mitochondrial genes, divided by total UMI counts multiplied by 100. Here we use MT. MT indicates those genes which related with the MT. If data belongs to human, we need to use a capitalize this MT. If data belongs to mice, we can use small. So you must be keep in mind. Let's run. Okay, it was I stopped because mistakenly it was typed A, so shows object A not found. So again we run this combined mitochondrial percent, this command, and now if we look after this command, we look the same metadata view. Now here is column added with the percent mitochondria. Before that, only N count and N features were available. If we want to check top 5 cells or first 5 cells, we can use this command, and these are the first 5 cells, and each cell by using this is the N count, RNA, N feature, and mitochondrial percentage. Let's okay, this is not related with RNA analysis, but it is just for my laptop because it's a little bit slow, so I use different ways to speed its speed, increase its speed. So just run it. Now because this data is a little bit complicated, we can not decide, we cannot judge, we cannot interpret data easily. So we want that this data should be in feature and in count. This should be a constructed should be shown in Violin plot. So we use a Violin plots, same these features, and we use 3 columns. Just we need to run this one. Okay, it's going on. So after Violin plots we can make scattered plots used to check a relationship between in feature and counts, and we can compare control and LPS, just like in my case. This scattered plots helps to visualize general trend, and just like if we look at this figures, uh, here in feature just like control and LPS, because of merging data, now it's easy for us to visualize two groups together in one figure. Our data indicates this is our data. We didn't do any filtrations, anything else extra. So our data indicates in features less than 6,000 and more than 4,000, in between 5,000, we can say, while in count near about 20,000, and while percent empty, if we look at the, it's near about we can say 5 to 6, or simply we can say 10. Now and this is just a violent plot of different. Now we want to draw scattered plots of these two comparison. Scattered plots is easy visualize general trend in cells that far away from the line, midlines, which indicates low quality cells. It can easily tell us about the quality, how our data, how our cells, how our data behaves by using a scattered plot. Let's run it. Okay, look at this. This scattered plot indicates both groups control and LPS, and this is the in count and this is the in feature. Our data moves along with a straight line, so it indicates the good quality of the data. Good quality of data should move with a straight line. So after scattered plot, and we also already visualize in feature and percent empty. So now we select we set filtering parameters or cutoff values for in features and counts. Now if we look at the cutoff values, normally cutoff values for in features selected, it should not be less than 200 to 500, because if genes expressed per cells less than 200, it indicates cell might be dead or low quality cell. It should not be more than 6,000. Sometimes it should be selected on 2500 or 6,000, or simply we can say 7,000 whatsoever. But before that, we already visualize our data indicates that in between simply we can say about 500 or 5,000. So in counts usually used with the in features, there is just for better resolutions, percent empty mitochondrial percent cutoff values depends on the type of cell. If cells are tissue, if tissue are healthy, then we can select a cutoff value less than 5 if tissue or cells under stress, tissue under stress. So just like tumor, so we can set a cutoff values 10 to 20. Here just like suppose we select cutoff values 200 in features and less more than 200 and less than 6000, while percent empty is our percent was 5 to 6. Simply we can select a 10, less than 10. Now let's run it. Now second step is normalization. What's normalization? Why it's important? Because if we cannot run the normalizations, gene expression raw data per cell might be vary between the cells. So if gene expressions vary between the cells, we cannot compare the cells. For the comparisons, it should be equal. So these variations might be occurs not because of biological difference, but it might be because of its sequence deeply. These variations leads to cause bias. So minimizing or decreasing these variations, we can use normalization functions which is available in Seurat. How it works? Normalizations, background of normalizations. This works by just like this formula. If a cell has a total UMI count 10,000 and gene A has 100 UMIs, so it works just like normally 10,000 has been set as a default, but we can change manually. This normal what happens, this normalized per cells then log transforms to compress large values. Again, this is not part of the single-cell RNA data-seq analysis, just for me to boost the laptop and increase speed and storage. Now let's combine. Now let's run normalization. Okay, now it's going on normalizing layers, just like counts control. After this counts control finish, then counts LPS will started. Just like normalization can be can be changed manually, just like scale factor can be selected as 8,000 or 9,000, 7,000. It depends on us. Now this part we are going to wind up here, just like normalizing layer counts control has been completed. Now counts LPS is going on. In next part we will discuss further. Take care of yourself, please, and wait for next part. If you want to actually I am just making a video for myself. In future when I forget anything, I may look my videos to again these things.